Good morning, everyone. Today, I'm giving a talk about the lymphatic system, and this week I also have another lecture with you that is still about the same area of the histology. Today, I will give you a kind of introduction to the field of lymphatic organs and histology of the lymphatic organs, and I will tell you some basic terms that we understand each other, that you understand the basics of the lymphatic system. We will talk about cells, of course. If it's about immunity, we have to discuss the cells which are part of the system related to the innate immunity. And I will also talk about the cells of the acquired immune system and or immunity. And I will very briefly highlight the internal framework of the lymphoid tissues. There, I don't want to talk a lot about. This is something that is just a recap again. And the next lecture is about the histology of the lymphatic tissues. Except the thymus, I will talk about the thymus gland today. The bone marrow we won't discuss in detail, and the secondary or peripheral lymphatic organs is the program for the next lecture. So this would be the overview for the coming lectures, or today and next time. Okay, then let's start with the basic terms immediately. So, what is the lymphatic system? If you want to define the lymphatic system, I think everyone knows that it's a kind of defense system that should protect the body, the organism, that we are not infected, or at least if we get infected, that we are protected against pathogens, viruses, bacteria, parasites can be pointed out. But of course, foreign bodies like splinters in your skin, if you work in the garden, for example, will be also removed or attacked by the lymphatic system. And a very important branch of the scientific research in direction of cancer research is the anti-cancer immunity. The tumor cells, cancer tissue is normally also removed by the immune system. So this is something that is one could define it as the lymphatic system. Next, I have to tell you in advance that this field is not easy. And we see many times that students have problems with this if they get these topics in the exam. And the reason for that is most likely that some structures we just don't see in the slides or in the microscope. We have, however, histological classes. We will see the sections, the slides, but many times the histological structure is that complex that we do not really see details there, which therefore we have to learn in theory. Also, you didn't have immunology yet, which is a subject where you will learn the molecular biology or molecular level of immunology. And because of this is missing, it's difficult to start with the introduction, and the time is always too short. And therefore, my goal is not to tell you everything about the immune system in 2 times 45 minutes, but I will highlight some key points, which is important for you if you want to prepare for the exam. And if it's that difficult, one may ask the question, let's leave it. Why to talk about it? We don't need this. I do not agree with that, because we have to study the immune system. You will be medical doctors, hopefully all of you will start to treat patients. And when you will be a young doctor, you will feel that you are the person who helps the patient. You will heal the patient. You will cure the patient. I have bad news for you. You won't. You probably will help the patient to survive until the immune system wins the fight against the disease. So normally the immune system is that which is dedicated to protect our body against the infections and other kinds of diseases. Therefore, the natural healing process is part of the life, and therefore we have to understand that, since we will be doctors, or you will be doctors. On the other hand, the immune system might go wrong, and sometimes the immune system is not effective enough. In some cases, people have hypoactive immune system, immune suppression, which means that they are not protected properly, sometimes because of an inherited problem. Sometimes we doctors give medications in order to reduce the activity of the immune system. For example, if you have a patient who received a transplanted organ, and we have to avoid the rejection of the implanted organ, we have to push down the immune system. And many people suffer these days from allergy. Many plants are growing now, blooming, and then the pollen, which is spreading everywhere, if you breathe them in, may develop symptoms, allergy. But sometimes the immune system attacks the body's own parts. These are the autoimmune diseases. Unfortunately, they are getting more and more common, and therefore you will meet these kind of cases for sure also in future. So this is why we have to study the immune system. If we would like to understand the system, one slide from my lecture, from my anatomy lecture in Anatomy 1, I took here because I have bad experience with this area in the exams most of the times. So what is the lymphatic circulation and how does that work is the next question. So the slide shows you here blood vessels, arteries, veins in blue, and capillaries. And you know that on the arterial side, the pressure is higher, and on the venous side, the pressure is lower. Because of this reason and because of other reasons, the vascular wall is most of the times permeable for water and for the extracellular fluid, and some fluid will go into the extravasal space outside the blood vessels, and this fluid returns, not the entire amount of the fluid, but the fluid in part returns to the venous circulation. Because of this reason, the fluid is transported by the veins away, but not completely. Daily, few liters of fluid stays in the tissues, and this has to be collected. And for this reason, we have the system of the lymph capillaries, and they collect the fluid. Now, if we have already such a complex extravascular system made for the lymph fluid, why not to put filters into this vascular system, the lymph nodes, which filter this lymph fluid? Because one can imagine that bacteria, viruses, pathogens, even cancer cells may enter the lymph capillaries, and therefore lymph nodes have to collect those pathogens in order to remove them from the lymphatic circulation, because the fluid comes to larger and larger lymphatic vessels, and finally, in front of the spine, there is a duct called thoracic duct, which drains the lymph fluid into the large veins on the neck, into this so-called venous angle. And the majority of the fluid returns on the left side. Some fluid also goes to the right side. This is something that you will study in anatomy, but it's enough today if you understand that the large lymphatic vessels, the thoracic duct, the right lymphatic trunk will drain into the large veins, and this is how the lymph fluid returns into the blood circulation. Therefore, it is essential that the fluid is filtered by the lymph nodes, that no bacteria or pathogens can enter the bloodstream. Okay, this is about lymph circulation, just a review. Theoretically, I have explained this for you in the last semester. Again, to the basic terms, there is a classification which I would like to ask you to study for the upcoming exam period. This scheme on the right side shows a quite simple overview of the lymphatic system, not a complete one, but most of the lymphatic organs are indicated. One can divide the lymphatic organs into two main categories. We have the primary or central lymphatic organs. One is the bone marrow already, that you already know from your last semester studies. I don't go into those details at the moment. And the thymus is an organ which still belongs to the primary lymphatic organs. The thymus is situated right behind the sternum in the thoracic cavity, And this is what I would like to talk about at the end of this lecture today. What I won't talk about today, but I have to define it, that from the primary lymphatic organs, the lymphatic cells will migrate into the secondary or peripheral lymphatic organs, and these are responsible for the immune reaction. We will talk about the lymphatic follicles next time. This is not indicated in the diagram on the right side, but the tonsils, at least in part, are here indicated. But you know from your anatomy studies already the tonsils in the oral and pharyngeal spaces. Lymph nodes I have mentioned, but you know many lymph nodes. The spleen is an important lymphatic organ, and not indicated on the figure again, but there are lymphatic systems associated to the body surfaces, like the skin-associated lymphatic tissue and the mucosa-associated lymphoid tissues. I will come back to these points next time, not today. But this is the classification of the lymphatic organs. We have primary ones and secondary ones. If I go to the primary lymphatic organs, just for a half a slide to remind you how it was. You know the bone marrow tissue from your last semester studies. This scheme summarizes how the cells of the bone marrow develop and what developmental stages of those cells are found in the bone marrow tissue. You know these colony forming units and also the erythrocyte, thrombocyte, neutrophil, and so on, how they develop. If you remember that, we discussed that time that the bone marrow gives rise also to the B lymphocytes and the T lymphocyte cells, the precursors of those, and also the natural killer cells come from the bone marrow tissue. So the cells of the immune system also originate from the bone marrow, besides those what you already know from your last semester studies in the blood smear of the peripheral blood. Then the T lymphocytes exit the bone marrow tissue, and they will travel and enter the thymus gland. And in the thymus, there is a quite complex differentiation process taking place, where the T lymphocytes have to go through. They proliferate here also, and they also divide. And finally, they turn into mature T lymphocytes. So the thymus is a primary lymphatic organ where the T lymphocytes develop. And then finally, these cells will then exit the bone marrow, and they will go into the secondary lymphatic tissue. So the mature lymphocytes, both B and T lymphocytes, will then enter the lymphatic organs I listed earlier as secondary lymphatic organs. They will communicate with each other with the cytokines. This is a specific group of hormone-like substances which will be responsible for the control of the immune reactions and functions. So this is how we classify the lymphatic organs, and in this scheme you can see both primary and secondary lymphatic organs. And the next basic term is the term of immunity. The word immunity comes from the Latin immunis, immune, which means to be freed, to be independent from something, or to be accepted from something. And everyone understands that the immunity means that to be protected against something. And one can divide the immunity into parts, two main parts. There is a phylogenetically older part that one can designate as the innate immunity. And in contrast, or in comparison to this, we have the acquired immunity, which is developing later in life. And also it has to be mentioned that the acquired immunity is sorry, somebody has the microphone on, please switch it off. So we have the acquired immunity that is phylogenetically more new achievement, and it has also a great significance in the immune reaction and immune function. Both we have Both are very important, but they show some differences, and I would like to compare them now. So the innate immunity is something that is not pathogen specific or not antigen specific. What does that mean? It means that it does not really recognize what exactly it is which enters the body. It only knows it is not our part, it is not our own tissue, so it has to be attacked, but it doesn't really recognize the individual bacteria or viruses, but can distinguish between own and foreign parts. But it has a rapid, very, very fast activation. So if you are working in the kitchen and you cut your finger accidentally, you know that if you cut your finger with a dirty knife, very soon you get a reddish skin decoloring because the immune system is there, gets activated, and an inflammatory process starts pretty fast, which accelerates. But the acceleration of this is a linear acceleration. I will come back to this point. The linear acceleration sounds good, but bacteria and viruses might grow exponentially. Therefore, an additional system was required. And this is the acquired immune system, which is in comparison to the innate one, already more complex, which means that it is already antigen specific. So it can exactly recognize and identify the individual bacteria, viruses, and pathogens if they are already known. But this is somewhat time consuming, so it will require a little bit more time until it gets activated. But if once it got activated, it will develop a memory, an immunological memory, which means that for years, for decades, the immune system may remember the individual pathogens. And if we meet the same bacterium or virus again, then the second reaction of the immune system comes really fast, and it will develop exponentially and accelerates really fast, that the bacterium or the virus, if the immune system works well, has no hope to infect us again. Like childhood diseases, you know, we can get those once in our lives if we have a normally working immune system. The immune system remembers and attacks the pathogen again. And if I got childhood diseases when I was in the kindergarten, and my son or my uncle when the children of my children will take this again to me at home, my grandchildren will infect me, then I will develop the disease, most likely not anymore, because my immune system remembers what it was. Okay, so this is how it goes with the immunity. Next, we will go to some recap. One can divide the cells of the immune system into groups. How can we do that? If we go back to the parts of the immune system and we split it into innate and acquired immunity, then one can say that we have the cells which we will classify into the innate immune system. These are the granulocytes, monocytes, macrophages. These cells you already have to know. Dendritic cells and natural killer cells you don't know yet, or you don't know that much about these cells at the moment. And on the other hand, we have the cells of the acquired immune system, and one can classify the B, the T lymphocytes, and the follicular dendritic cells into this group. I will talk about the follicular dendritic cells a bit also today and next time. So let's review these a bit and talk about these a bit. So what are the cells of the innate immunity? This is already well known for you. A blood smear stained by white Grünwald Gimsa, and here you can see granulocytes, neutrophilic ones, eosinophilic ones, and those rare basophilic granulocytes. You know everything about these cells. If not, it's time to review. What I would like to point out is that neutrophils have a very strong phagocytotic activity. They may eliminate bacteria, and they also produce several toxic molecules which will be able to eliminate the bacterial infection. Eosinophilic granulocytes are more efficient to attack parasites, which infect us a little bit more rarely. However, it might happen. also in the allergic reactions, eosinophils have some significance. Basophilic granulocytes are not very common in the peripheral blood. You know these counts for sure. If not, you have to review them, but they might have a regulatory role and also some anti-parasitic and antibacterial functions by producing toxic molecules. Please review the granulocytes, what you know about those from your last semester studies. Monocytes, macrophages belong to the innate immunity again. This picture shows you a monocyte in the peripheral blood. You can see this little bit half moon shaped or indented nucleus here with some hetero and euchromatic nucleus. And also you can see many little bubbles within the cells, which proves that the cell removed something from the bloodstream. It performed phagocytosis, and it ate up some bacteria or viruses or particles. So these cells have a very strong phagocytotic activity. In addition, we have to point out that they are responsible for antigen presentation. And the antigen presentation is something that is essential to know, because what does that mean? Because it's a very important function of the immune system. So very briefly, the phagocytotic activity means that the cells may eat up bacteria or viruses. The cells will then pick up the virus or the bacterium and they digest it. Now, they do not break down completely the proteins of those pathogens, but they leave them in small particles in few amino acids peptide chains. And they will put these remnants of the proteins of the bacteria onto the surface of the cell. So they present the antigens, the immune markers of the bacteria and the viruses which they removed, they eliminated. So they are able to show for the other immune cells that these are the bacteria, these are the viruses which attacked us. Therefore, they have a very important regulatory role in the immune system. Besides antigen presentation, they also produce cytokines, which are very important messengers to attract the other immune cells to the place of the infection. You might remember the monocyte count and also the size of these cells. I don't want to review that. This is basic knowledge already. So these cells you must remember from your last semester studies in the peripheral blood. That time we didn't discuss the dendritic cells. They develop from the bone marrow also, and they perform phagocytosis. They also have antigen presenting and cytokine producing actions, but they are localized to specific organs. In the lymph nodes, you will find them. You will find them also in the spleen and also even in other organs. What you might remember, the Langerhans cells in the skin, in the epidermis of the skin, were also phagocytotic cells, and they are also a member of this group because they perform antigen presentation. So they will show the bacteria, the viruses, the pathogens for the other immune cells, and they present those. Next, this figure shows you the surface. It's a scanning electron microscopic picture, which is colored. It's very wavy, uneven, irregular surface here is characteristic for the dendritic cells. This is the reason why they are called dendritic cells, because they have processes like tree-like branching processes, dendros in Greek, the tree, and this presents the antigens for this lymphocyte here, for example. Natural killer cells also belong to the innate immunity. They belong to the cells of the immune system which are very ancient. It's a very ancient type of lymphocytes. They have a very aggressive way to treat the cells which they have to eliminate. They perforate the cell membranes of other cells. Typically, they are named as the most important cells against anti-tumor immunity, against cancer, but other infected or virus-infected cells may be also eliminated by natural killer cells. It's very important. It's a very important branch of cancer research. How to bring the natural killer cells to remove the tumor tissue, because cancer is something somehow also a failure of the immune system, that the immune system does not remove the cancer cells efficiently enough. And if we could somehow force the NK cells to do that, we would potentially be able to treat also the cancer. So this picture shows you a tumor cell here, and it is attacked by a natural killer cell and perforates the cell membrane and kills it. Okay, and now let's turn to the cells of the acquired immunity. Here I have to start with the B lymphocytes. The B lymphocytes you might remember in the peripheral blood, but it's really difficult to distinguish B and T lymphocytes from each other. We have to do a immunohistochemistry in order to distinguish those cells from each other. So to be honest, I really don't know if this cell is really a B lymphocyte or a T lymphocyte, but one could do an immunolabeling and then you would be able to distinguish. But anyhow, you have to know about B lymphocytes that they occur in lymphatic follicles. We will talk about these next time. And also in the peripheral blood, they show up. And what you have to know about these is that they are responsible for the humoral immunity. What does that mean? That means that for many, many decades or many years, we know that the fluid of the human body, fluids of the human body, has a quite strong antibacterial feature. And people call this humoral immunity because they knew that the fluid has it. Now, we know that the most of the body fluids will contain antibodies, and these antibodies are proteins which can recognize and can bind to the pathogens, and they can mark them, and some of those can even eliminate the bacteria or the viruses. And therefore, the B lymphocytes are the key cells of the humoral immunity, because they develop into plasma cells, which look a bit differently. And these plasma cells are then responsible for the production of the antibodies. They are major differentiated B lymphocytes. They have a typically eccentric nucleus. They are large cells. They have a very strongly basophilic cytoplasm, because if we zoom in with electron microscope, one can find a very strongly developed rough endoplasmic reticulum in the cells, since they produce proteins for exocytosis. Also, it's good to know that the cells have the radial pattern of heterochromatic nucleus with some euchromatin. It's a radial pattern of heterochromatin characteristic for the plasma cells. They can be found in the lymphatic organs most of the times. The other type of the acquired immune cells is the T lymphocyte. I mentioned that they develop in the thymus. They might be found in the spleen, in the lymph node, and in the blood. What these terms mean, I will highlight next time. Periarterial lymphoid sheath and the paracortex are spleen and lymph node areas respectively, but we will come back to this point next time. Now, just I don't want to go into the complete classification of the lymphocytes because it's a very complex issue, but one has to mention that we have the so-called CD4 and the CD8 positive lymphocytes. See, these are the clusters of differentiation markers on the surface of the cell. One can use specific stainings to mark these surface molecules, and this way you can distinguish the kinds of cells from each other. One subtype of the lymphocytes called helper cells carry the CD4 molecules, and they are very important regulatory cells. They will kind of control the entire immune system, and they will be the kinds of communication centers, and they will control the action of other immune cells. Therefore, it is a key point of the immune system, and if these cells die, they don't work properly, we have some immune deficiency. And this is exactly what the human immune deficiency virus does, that HIV attacks the helper cells, the CD4 positive lymphocytes, and kills them. Therefore, the patient develops an acquired immune deficiency syndrome because the acquired part of the immune system is missing, since the communication is destroyed in the system. Like in modern wars, people don't want to kill each other in the war anymore. It's enough if people destroy the communication centers and the war is over, because people cannot communicate with each other anymore. And also, the other type of the lymphocytes, the T lymphocytes, are important for us. This is the CD8 positive group of lymphocytes, also called cytotoxic T lymphocytes. They are the cells which can eliminate virus-infected or even cancer cells. They also have a great significance in the immunity. You know that the viruses develop within the cell, they grow there, they replicate there. And if you want to eliminate a viral infection, you have to eliminate the virus-infected cells. The problem is that in some cases, the lymphocytes remove cells which are really important for us. For instance, if somebody suffers a hepatitis infection, hepatitis B, for example, is a viral disease, and if the liver is infected, the immune system will attack the liver, and the patient may even develop jaundice at the end because the liver cells will be removed together with the virus. So the immune system may go too far in some cases. But the T lymphocytes have an extremely important role in that. Okay, just a review on the lymphoid tissues, which is a slide that you might remember from your earlier semester studies. It was a slide we taught in the first semester also. It's about reticular connective tissue. And why I'm taking this here is that this is the system which forms the framework of the entire immune system, except for the thymus. The other lymphatic organs, including the bone marrow, the spleen, the tonsils, you always find this kind of internal framework which supports the tissue. It is made up by reticulum
I'm
cells.
sorry.
Yes?
Is it this teaching recorded by someone? Because
I do record it.
okay, and where you will upload it? In team files or?
Yes, I will load it up after the class into the Teams. There you will find you will find a place on the top of the page called Files, and we will load it up. Okay.
Okay. Thank you.
Okay, so this system is a network of reticulum cells. It's a kind of connective tissue cells, which is supported by reticular fibers. Also, it's a kind of framework. And in the system, in the cavities of this framework, you have here many cells, lymphocytes, which can be found here. These are round, smaller cells, and those star-shaped cells with elongated processes connected to each other form this framework, which supports the entire system. So this is how the reticular connective tissue looks like, which supports the system. It's hard to see in the histological preparation. One has to wash out the lymphocytes in order to be able to study the cells, because they are covered normally by the lymphocytes as they are in a really high number there. But once you do a histological technique that gets rid of the lymphocytes, you can basically show the cells much more easily. So don't forget that most of the lymphatic organs have reticular connective tissue inside, except the thymus. Okay, and at the end of this lecture today, what I would like to go through is the histology of the thymus, and I will then next time go on with the histology of the secondary lymphatic organs. But let's go on and let's talk a bit about the thymus. So in the first part of the lecture, I showed you that there is an organ behind the sternum. And it's just not just an accident that I took this really awful picture. This is the chest cavity of a baby, and if we open the chest cavity of a baby, one can find a huge organ behind the sternum. This is the thymus, and you can see that proportionally it's a big organ I don't say it is as large as the lungs are, but it's almost comparable in size with that. It's above the heart, it's above the pericardium and in front of the large vessels in the chest cavity in newborn babies. If we would study the organ in the embryonic life, it starts to develop as an epithelial structure. You will learn in the embryology lectures how it develops from the pharyngeal pouches as an epithelial organ. And very soon, lymphatic cells will migrate into the thymus. At this stage, you call it lymphatic thymus. This developmental stage occurs in the 3rd embryonic month, and it stays until puberty. And after puberty, the fat cells will start to replace the lymphocytes. Sorry, and finally, the adipose thymus develops at the end. In some people, the lymphatic tissue may persist. In this case, you call the situation persistent thymus, but we believe that this is not completely normal. If we look at the curve of the different tissues in relationship to the time, one can see how the proportion of the tissues will change, and with age, how the fat will accumulate and how the lymphatic tissue will decrease its proportion within the organ with the course of aging. Before birth, basically the lymphocytes will be characteristic only. If we look into the microscope with a low power magnification, one can find here lobes and lobules. These are little units, each of which is divided into a cortex, into an outer unit, which is a little more rich on cells, and in the center you have the medulla here. As I mentioned earlier, we can find in the organ T lymphocytes, dendritic cells, macrophages, and as I mentioned, in contrast to the other lymphatic organs, you have epithelial reticulum cells. Please do not mix this up with the reticular connective tissue. There is no reticular connective tissue here. No reticular fibers occur in this organ. And also, please note that there are no lymphatic follicles in here. I will come back to this point in the next lecture. What does that mean? The functions I mentioned already, that we have bone marrow derived T lymphocyte precursor cells here, which proliferate and differentiate here into functionally mature T lymphocytes. And after this process has been fulfilled, then the cells may exit through specific veins at the border between the medulla and the cortex, and they enter the bloodstream. If we go into the medulla of the thymus and we zoom in, one can find these larger cells with a big nucleus, and there is a nucleolus in the center of the epithelial cells, also recognizable. This is the framework of the organ. These are the epithelial reticulum cells. The epithelial reticulum cells produce different kinds of cytokines like thymosin and thymopoietin, and these will then control the growth of the lymphocytes here and the differentiation of those as well. But the epithelial reticulum cells have a turnover, and at the end of their lives, they will approach the center of the lobes, and this very specific structure they form, which are called Hassall's corpuscles. Hassall's corpuscles are structures made up by epithelial reticulum cells which are degenerating. You can see here these keratohyalin granules, showing that these are epithelial cells undergoing a process which is similar to the keratinization that you might remember from your first semester studies. And finally, they degenerate and they die. We don't know that much about the Hassall's corpuscles. Their function is unknown even nowadays. We do know that they might have a function as their number and size changes in some autoimmune diseases or in some immunological problems, but we really don't know what they do yet. So this is found in the center of the medulla. If you see that, you can be sure it's the thymus, because this is the only place in the human body where you can find these Hassall's corpuscles. They help to identify the slide if you want to identify them. There is one term which is sometimes a problem in the exam, and therefore I would like to highlight that also. These are developing lymphocytes here, and this is a blood vessel. In the blood vessel, there is always blood, and in the blood, bacteria and viruses may be present. But it's very important that in the developing lymphocytes do not meet too early the pathogens. They have to be protected. They have to be separated for sure from the pathogens, that they do not tolerate those. So they would develop a tolerance if they would meet them too early. But in order to avoid that, there is a very strong barrier here between the blood and the developing lymphocytes, which has layers. So this is a capillary which has an endothelial wall that has a basal lamina, and you have some connective tissue around the blood vessel, and then the epithelial reticulum cell comes here, which also has a basal lamina, and these layers together will separate the bloodstream from the developing lymphocytes in order to avoid that the bacteria and the viruses can meet the lymphocytes during their development, because that would be too early and they would develop a tolerance. And my final slide is about a process that would require alone a lecture. I don't have time to talk about this in detail, but in immunology subject you will hear a lot about this. But that much you have to know also in our exams, that the Thymus is there to help the selection and the development of the lymphocytes, and there are two steps they have to pass in order to develop into normal functioning lymphocytes. The first step is a positive selection, which means that lymphocytes are produced, and during this production, on the surface of the cells, their receptors may change. And the question is if these cells are able to recognize these so-called MHC1 or MHC2 molecules. This is the abbreviation of the major histocompatibility complex 1 and 2. So this is a kind of ID card for the cells. If a cell carries that, one can decide if the cell belongs to the body or it doesn't. So if a lymphocyte is good enough, it is able to distinguish and to recognize the MHC1 or MHC2 molecules. Those lymphocytes which are unable to do that will be unable to survive. Yes?
I have a question. MHC1 is a part of the antigen, which is a part of antigen, should be recognized by lymphocyte?
The MHC1 molecule is also an antigen, yes. But the question is now if the lymphocyte is able to recognize the MHC1 by the T cell receptor. But I don't want to go into the details now because there is no time for that, and you also don't have to know the details for our exam. But in the immunology subject, you will learn about that more detail. The MHC molecule is something that identifies the cell on the surface, if the cell belongs to our body or it's something else, or it's an abnormal cell. Okay? This is about MHC1 and 2. So this is the first step of the positive selection. And those cells which are able to recognize the MHC1 or MHC2 molecules can survive. Those which, however, are unable to do that will be eliminated by apoptotic cell death. Then those cells which pass this first positive selection are tested again by a so-called negative selection. In this case, the cells are checked, the developing T cells are checked if they are able to recognize the MHC molecules in the neighborhood of antigens which may belong to our own body or may come from foreign sources. And in this case, it's really important that the T lymphocytes can distinguish the foreign proteins and our own proteins. And those cells which react with high affinity with the proteins which come from outside the body, they are required for distinguishing or for recognizing the pathogens. But those cells which have a high affinity to bind to our own antigens, they cannot survive. They are not allowed to survive because they would attack our own body, our own antigens, our own cells, and therefore they carry the chance of an autoimmune disease. Therefore, they are eliminated. So and again, a selection occurs, and those cells which have high affinity to our own antigens, they will be removed, and they undergo apoptosis again in order to rule out that the lymphocytes can attack our own body. So this is the second step of the T lymphocyte selection. There are two steps. Much more details you will study in your immunology subject. So I think the time is over, so I don't want to take your day away. Next time I will go on with the secondary lymphatic organs, and as a final picture of the lecture, just a nice photo from the Lake Balaton in the autumn. Thank you for your attention.